Great. Hello, everyone. So I'm going to get started. My name is Brian O'Connor. I'm the Technical Director of the Analysis Core at the UCSC Genomics Institute. And today I'm going to tell you about our large-scale cloud-based analysis of cancer genomes. In particular, I'm going to tell you about the lessons that we learned from the PCAWG project. So just as an overview for my slides, I'm really kind of dividing things into three parts, with PCAWG being the past and sort of lessons learned on how to do large-scale genomics analysis on the cloud. And I'm looking towards the present in terms of translating what we've done into tools that other people can use. And finally, I'm talking about the future through the Global Alliance and establishing standards by which we can have other people use similar infrastructure and have that be compatible with each other through the GA4GH. So just as a background, the PCAWG project is a large international collaboration, so it stands for PanCancer Analysis of Whole Genomes. It was organized by the ICGC, and it comprises of approximately 5,800 whole human genomes that corresponded to about 2,800 cancer donors, 1,300 of which had RNA-seq data. Now, the goal of the technical working group was to consistently analyze this data set. It came from a variety of projects. They used different workflows. So we wanted to enable scientists to do an awesome analysis on this by having a consistent alignment and variant calling, somatic variant calling pipeline applied to this data set. The challenges for this project were, its scope and its geographic distribution. This was not a funded effort. This is actually a collaboration of volunteers all around the world. About 700 scientists were involved in this project. And that meant we had 8 different sites to store the data through a system called Genomes, which you can think of it as a multi-server SFTP server. We peaked at about 900 terabytes with this project. And the biggest challenge for my team and I back at OICR was building the infrastructure that could run the consistent analysis pipelines across 14 cloud and HPC environments simultaneously. 3 of those were commercial, Amazon being our go-to cloud environment on that side, 7 OpenStack environments, and 4 HPC environments. So at the peak of the project, we were managing about 630 VMs across these environments that corresponded to about 15,000 cores. So it was a very challenging infrastructure project for us. In terms of the pipelines themselves, we were trying to establish core analysis pipelines that included BWA-MEM alignment to a standardized genome, and then best practice pipelines that were created with our team and the team at Sanger, DKFZ, MBOL, and the Broad for calling somatic variants. These variant calls would then be filtered and merged and consensus called, and that consensus calling process was validated through a 63 donor validation data set. And further analysis was then done downstream of the filtered, merged, and consensus called variants. That process was really in full swing by summertime, and now we're in the state of actually working on publications from this project. So it's a very exciting period of time for the PCAWG project. From the technical infrastructure side, the challenges of spreading our analysis across so many different environments, we learned several key lessons that I think are interesting and informative of our future work. The first one was a lesson about cloud policies and how those can really shape the capabilities of what we can do in clouds. The second was about how to make tools portable when we have so many environments to work in. The third was about how to make the execution platform distributed and fault-tolerant. And finally, we learned a surprising lesson about commercial cloud costs. So on the first side, going into this project, TCGA data we could not place on the commercial clouds. ICGC data we could only transiently process. But things started to change in March of 2015. The NIH updated their dbGaP cloud policy that allowed us to use commercial clouds for processing data. In May of 2015, the ICGC DACO updated the ICGC cloud policy. And that allowed us to not only compute in those environments, but also redistribute ICGC data, which comprised about half this data set, through commercial cloud environments. It enabled really cool partnerships here with Amazon, the Public Datasets program, allowing us to redistribute PCAWG data through Amazon S3. And that enabled our partners with 7 Bridges and DNA Nexus that provided compute for this platform to access that data within that cloud environment. So that was a small thing in terms of a policy change, but it really shifted the way that we did our compute on the project. So originally, we would look at a standardized sort of single centralized metadata index, figure out what needed to be run across our fleet of clouds, in-queue work in those clouds. And then if we were working in Amazon Cloud, spin up spot instances that needed to pull the data from these external Genomes repositories, these external storage repositories running around the globe. And that was not ideal because that pipe was limited. So we could only pull in so much data to process in Amazon. When these policy changes were put into effect, we suddenly could actually host ICGC data within the Amazon environment. We could load it into S3, and that allowed us to pre-cache data into that environment and then do much larger scale compute on that data because of the scalability of S3. And that also allowed us to share within the cloud environment with DNA Nexus and 7 Bridges. So a great shift for us in that project. It was really something that changed the way that we worked. Something else that had a really amazing impact on the PCAWG project was really seeing Docker released as a 1.0. About halfway through our core analysis on this project, Docker released its 1.0, and we felt comfortable using it in this project as a key component. What we ended up doing is moving to a model where we moved away from a model of building worker nodes on demand using Ansible playbooks to actually set up a worker node from scratch, install all the tools on it, install the workflow. That was a very time-consuming process that was very error-prone. We moved instead to making portable Docker images that contained everything in the kitchen sink. So we included workflow definitions, config files, some of the reference files, all the tools that we needed, and we covered the core pipelines. This was BWA mem alignment and the 3 somatic variant calling pipelines from Broad, DKFZ, Emble, and Sanger. And so this was really transformative for us because suddenly we could move around these lightweight Docker containers. These Docker images had everything that we needed in them. It really cut down our provisioning process. It really improved the reliability of that provisioning, but also it allowed us to start exploring other environments for compute, including handing these to HPC environments that were participants in PCAWG and having those Docker containers run there as well. So the third lesson that we learned is really, building and running infrastructure on the cloud is different. You don't want to treat it the same way that you've worked previously. So going into this, a lot of the academic folks and myself had more experience with HPC. So our first sort of architecture for running analysis within PCAWG was really setting up something that looked like a traditional HPC cluster. We run Sudgren Engine, we had Gluster running, we created these little mini clusters across the clouds and scheduled out work to them, as you sort of typically do. What we found, though, is in environments like Amazon, where we're interacting with a spot marketplace and we'd lose nodes, or OpenStack environments that were created for PCAWG and actually new environments, so they're still having sort of teething issues. These environments were not amenable to running SGE and having SGE lose nodes. So instead, we kind of shifted halfway through to an architecture where one VM processed one donor, ran one workflow, and at least that was a self-contained infrastructure. But we weren't particularly happy with it because we didn't really want to work in that way. We want to work in a way that was much more nimble and able to deal with failure on its own rather than having us reschedule work that failed. So we ultimately, in conjunction with moving to Docker-based tools and workflows, we built our architecture, what we called Architecture 3. And the hallmark of that was each of the cloud environments we worked in, including Amazon, had a queue. And we would in-queue the work that needed to be done in that environment. And that queue was read by a provisioner that would spin up VMs. In Amazon, it was spot instances. And those spot instances would have a worker daemon on them that would dequeue work and run the particular Docker-based workflow for that particular sample. What that gave us is a system that if workers disappeared, it automatically knew how to re-in-queue work so that we could be very robust to failure, whatever that failure was. All right, so the fourth lesson that we took away from PCAWG that actually turned out to be something that surprised most of the technical working group was really doing a sort of look at our expenses, looking at the institutes that built out OpenStack environments specifically for this project, and look at the cost of running on Amazon. So we used Amazon quite heavily in this project, especially early on, to bootstrap our way into running. We could get running on Amazon much more quickly than any of the custom-built OpenStack environments. So we had a lot of information about the true cost of running this analysis on Amazon using the spot marketplace. And we had a pretty good idea of what the costs were associated with building out over the course of a year and a half or so to 2.5 years was the entire lifespan of the project. We had a pretty good idea of the cost of building out our academic OpenStack cloud environments. So there's an interesting piece in Nature in 2015, Data Analysis, create a cloud commons. Encourage you to take a look at it because this figure is obviously too small here. But the take-home point is when we did the math, we looked at building out infrastructure, staffing it up, keeping it supported, and compared that to running the same pipeline, the Sanger pipeline, on Amazon. We actually had an order of magnitude difference in the price. Amazon was significantly cheaper for us to do compute on. So that was surprising. At the end of the day, when we looked at the actual cost of us running this analysis on Amazon, it boiled down to only about $62 per donor. And that may seem expensive to some, but put in the context of the $3,000 to $4,000 required to actually sequence these samples, it's a drop in the bucket. So we're actually very pleased with the cost savings and the scalability, the ability to spike our usage in Amazon that was afforded by this cloud. All right, so what about the legacy of the PCAWG? We learned a lot of great lessons. A lot of scientists got to use cloud environments for the very first time. We saw that it was a viable thing for analyzing cancer genomics data in the public clouds. We're in the phase right now where publications are coming soon. So this is a very exciting phase for the project. In terms of a legacy for the project, beyond just the publications, the Amazon AWS Public Datasets program has allowed us to redistribute the ICGC PCAWG data through S3 on the Amazon cloud environment. And I think that's really transformative. That and the NCI cloud pilots kind of give us a glimpse of the future of what it's going to be like to work with large-scale datasets, large-scale cancer datasets on cloud environments. So this is great for a researcher because I can come in, I can identify the data and get a token that allows me to access it from the DCC portal, dcc.icgc.org. And then I can go onto the cloud environment. I don't have to worry about downloading the data from EGA or SRA or some other repository, which many of you know might take weeks or months to do. And I don't have to worry about the storage cost of storing the BAM. I can just get to my actual compute on it. So I think those are very powerful sort of legacy items from PCAWG. In terms of what are we doing right now, between my group at Santa Cruz, between OICR, between a lot of other people that have contributed to the technical working group on PCAWG, the focus for us is to get our workflows that we used in PCAWG shared. Obviously, we want those to be part of our publications. But on top of that, we want to take the tools and infrastructure that we built for PCAWG and make that something that other people can use as well, that other large-scale projects or even smaller-scale projects can take advantage of. So I think there's 3 key tools from PCAWG or inspired by the work of PCAWG that I think are generally useful and things that many people will find useful. And those are Redwood, which is our file storage system on top of S3. That's Dockstore. It's our platform for sharing Docker-based tools and workflows. And Toil, which is coming out of the Santa Cruz group. This is our workflow execution platform that goes beyond the sort of compute that PCAWG did. So what I'm personally working on right now at Santa Cruz is taking all of these components that we built or were inspired by PCAWG and linking them together into a larger ecosystem. What we're doing here is building a system that by stringing these tools together, we can automate the process of data coming into the Institute. And so it starts on the left-hand side, where at the bottom, people are submitting data to us. These are our collaborators. They're submitting data to us through an upload client that writes it into the data storage service, aka Redwood. From that, we've established a metadata standard and indexed that metadata standard to build a donor-oriented document that makes it very clear what analysis has and has not been done on that particular donor. And from that, we have an action service. Currently, we're using Luigi for this that queries our Elasticsearch metadata index figures out what work needs to be done, and then enqueues that work in our queue, which calls Toil, to actually do the workflow execution. So this system altogether is able to process data and hand it back to our collaborators, hand it back to other scientists at the Institute. And in terms of the actual tools and workflows, we're placing those on Dockstore to get the greatest sort of benefit from other people using those tools as well. So just to dive into the details of the actual projects that I'm talking about here, the Redwood system is a scalable storage system built on top of S3. It is based on the ICGC storage system that powers the PCAWG data sharing through the Amazon Public Datasets program. In this client, it's quite different from the way that we worked in PCAWG originally, where we actually had these SFTP-like servers that would actually handle the entire transaction and entire transfer of data to a worker node. In this system, the client running on an EC2 host will simply use a token to request a particular blob of data, essentially. And that storage service that then authenticates that request will hand back a signed URL. And so the outcome of that is, whether it's the client is uploading or downloading data, is the actual large data transfers coming from S3. And what does that mean? That means it's a much more scalable system than the approach that PCAWG took that actually bottlenecked in many ways on the storage transfer. Since we're doing our heavy lifting coming directly from S3, it means I can throw hundreds of simultaneous clients on hundreds of nodes, hitting this and not see an appreciable decrease in the transfer speed. And this is an example of that. As I'm adding more client nodes that do simultaneous downloads, I'm not really seeing a degradation. Of course, this is why we love S3. We love the fact that it can handle so much data. In terms of workflow sharing, we really loved the approach that we took with Docker. We really thought that helped the project, that helped to make our tools and workflows much more mobile. But one of the issues that we ran into is we didn't actually have a standardized way of saying this Docker image has this tool in it, it takes these parameters, and this is how you run it. That was something that if you go on Docker Hub or Quay.io, right now you basically read a README and you figure out how to run it. We needed some sort of standardized approach where we could have a standard that would describe how to run this scientific tool. And so for the Dockstore project, which is formalizing our sharing of Docker-based tools and workflows, we embraced the CWL and WDL projects. So this is common workflow language and workflow definition language, respectively. CWL is its own open source project. WDL is coming out of the Broad. And what these descriptors allow us to do is have a very clear human and machine readable format that tells us what the inputs are, what the outputs are going to be, how to construct the command. So we've essentially, through Dockstore, standardized the way that we describe tools and workflows in Docker images. Now, as an academic project, we didn't want to reinvent the wheel. I'm sure almost everyone in this room actively uses GitHub and Bitbucket for storing source and maybe storing a Docker file. I'm sure most people in this room use Quay.io or Docker Hub for actually building the Docker images. So that's exactly what we want to do on Dockstore. We want to leverage those with Dockstore acting as a registry that brings that information between Quay and GitHub together with that descriptor that standardizes the way that we can describe how to run that tool or that workflow. And so what we're doing with GitHub is we store the Docker file, we store a dockstore.cwl or a dockstore.wdl, and that Docker file is then built by Quay.io, which builds the Docker image and hosts it. And Dockstore is really bringing those two things together in order to have a common view of the content of GitHub, the content of Quay.io, and that descriptor itself. So where are we at with Dockstore? I'm really, really happy to say that Dockstore has actually reached its 1.0 release. So this is a production system. Multiple groups are using it now to exchange Docker-based tools and workflows. It supports a GA4GH standard that I'll tell you a little bit more about at the end of the talk. And it also supports the latest CWL. CWL itself is a project just released 1.0. We support 2 entity types. We support Docker-based tools, and we also support CWL and WDL native workflows as well, which are modeled as documents. And right now, the PCAWG group is really working hard to make sure all of our content, including the core pipelines, are being ported and available in Dockstore. We have all of them except the Broad at the moment. So in terms of what the Dockstore looks like, you can look at it today. You can use it today. It's in production, dockstore.org. The site has pretty much all the functionality that you need through the site itself, including search and tool management. If I dive into a particular page, you can see that it describes information about the container. If the font was a little bit bigger, you could probably see that it's giving you information about who made this container, when was it made, links back to GitHub, links back to Quay.io, and description, discussion, sharing links. But something that's the most important thing here, one of the outcomes of standardizing on CWL or WDL, standardizing the way that we describe how to run the tool, is it means that every tool or workflow that you find on Dockstore has a common methodology of executing it. So you always construct the command the same way. This makes it so much easier to find tools and execute them in a consistent way. To facilitate that, we have a very lightweight command line tool. If you find something that you like on Dockstore, you want to run it on your machine that supports Docker, this command line tool will help you to provision your files from standard URLs, pull the Docker container, execute the CWL or WDL wrapper to essentially create a Docker run, and then provision the files back out. So this is a simple mechanism for you to use something today that you find in Dockstore. What we're trying to work towards in the future is we don't want to be the only execution platform out there. We actually want 7bridges and Cureverse and other commercial platforms, as well as open source platforms like Galaxy and Consonance, to be able to pull and use tools and workflows from Dockstore as well. So we hope that in the long run, our goal is to have Dockstore tools runnable in a wide variety of platforms that allow you to scale up. So as a scientist, another feature that I think is really important about Dockstore, something that came out of the PCAWG project, is very, very clear versioning being extremely important. If I have a large cohort of data that I've run on a previous version of the pipeline, I may have new data that I want to bring in and use with that same version of the pipeline. So Dockstore provides a very clear link between the GitHub codebase, the Docker image hosted on Quay or Docker Hub, and the descriptor that describes how to run that particular version of the tool. And this enables you to find older tools, older versions of tools, and recreate or supplement existing data sets. It also has, again, bringing together the content of GitHub and Quay or Docker Hub. It's bringing together a single place that you can understand what the Docker file contains, how that particular tool was built, and also the descriptor itself, like how the tool is described in WDL or CWL, what its inputs or outputs are. And finally, as a developer, if you follow our SOP, if you use GitHub or Bitbucket, if you use Quay.io, which provides us a nice API, we can automate the registration of content from your Quay.io and GitHub account through the site. So if you followed our development SOP, which is a very lightweight SOP, you can basically gain the advantage of going into Dockstore, seeing your content that was built on Quay, and clicking a button to publish it. All right, so what's coming soon to Dockstore? We have several new features coming up. I think one of the most important is integrating the concept of public test data sets. So if you find a tool on Dockstore, I want to be able to run it with known good data, see examples of what the inputs and outputs should be. The other thing is we're actively working with commercial and open source providers to enable execution of tools and workflows from Dockstore. That is an ongoing process that we're actively working on. And we're also looking at things like signing Docker containers so we have a chain of trust, and cross-site indexing. So if we have many copies of Dockstore running around the world, we want to be able to cross-site index them and show their content across the network of Dockstore instances. So hopefully that gives you an idea of some of the storage system that are kind of coming out of the PCAWG project, the Dockstore.org, which is coming out of our sort of standardized way of packaging tools from the PCAWG project. I want to talk a little bit about how to do more efficient compute on AWS than what we did with PCAWG. So PCAWG was remarkable in being able to run in so many different environments, but we really didn't explore to great detail the way of making those individual workflows more efficient. And Toil here is a response to making individual workflows run more efficiently at a lower cost. In PCAWG, we ran everything in the kitchen sink was put into a Docker image, one VM, one Docker image running, one donor was processed on that image. We want to do something more efficient where we can actually scale out individual jobs from a workflow and grow and shrink the cluster on demand in order to increase our turnaround time for samples. So the Toil system is really built with that. It's built with large-scale, efficient work on Amazon as the key sort of driving feature. A recent recompute saw us managing a 30K core cluster and processing about 20,000 RNA-seq samples. So we're showing that we can actually do this sort of fine-grained job granularity, fine-grained execution efficiently, robustly, and at scale. So to give you some more details about what makes Toil unique and special, some of the more advanced features, unlike PCAWG, which had very static workflows, Toil workflows can actually be dynamic. The workflow can change during execution. This is something that not many workflow execution or workflow definition languages can support. Another thing that I think really makes it stand out is its ability to execute services for a given workflow. And the common example that we use quite a bit at Santa Cruz is executing and creating a Spark cluster for the execution of workflows that contain Atom steps. So we're using Atom here to speed up our BAM manipulations in some of our pipelines. And it's taking advantage of the fact that Toil knows how to spin up a Spark cluster in order to satisfy the needs of those jobs. From a user perspective, a lot of people like Toil because it's just pure Python, very, very simple API. You have all the functionality of the Python programming language. You can see here we're just defining a very simple task. We're assigning the word that we want to echo, basically. And we're telling it how much memory, how many cores are required. So we're giving those consumable resource requirements to our job and then telling Toil to execute this miniature one-step workflow. So it's a very simple syntax, but very powerful because of its underlying support of Python. In addition, we're supporting various environments. In production, we're using Mesos workers on AWS that are provisioned in an auto-scaling capability with Toil. But other batch processing systems have been submitted by the community, including SGE, LSF, and Slurm. So finally, I think that the key underlying, most important sort of pieces of Toil are its scalability and robustness. So one of the ways that we've made it scalable is the job store. This is the service that tracks the jobs that are completed and also intermediate files that are completed. This is designed to handle many concurrent workers. On top of that, we're using Mesos as our actual workers, and we've shown in testing that we can scale out through the auto-scaling code of Toil. We can scale out to about 50K nodes on Amazon EC2. So that's quite, I think it gives us quite a bit of headroom for scaling out our computes. And the workers themselves have been written in such a way to really reduce round trips to the central master. They're capable of doing their own scheduling on a node. They're essentially able to batch up scheduling to reduce that interaction with the centralized master. In terms of robustness, I think what really convinced me that Toil is a cool project is seeing the ability to take a Toil cluster, absolutely destroy it, and then bring it back online and have it pick up where it left off. So individual jobs are checkpointed upon completion. This is fundamentally different than what we did in PCAWG. If a given VM running a given Docker-based workflow for a given donor failed in PCAWG, we had to start from ground zero. In this system, if particular steps fail because of a VM going offline because of spot instance prices, for example, the system is smart enough to checkpoint the previous jobs and resume where it left off. So that's very, very powerful in an environment where nodes may come and go. The other thing that I think is interesting here is the Toil job store is designed to work in a variety of contexts. We primarily use it in Amazon with S3 and SimpleDB backing it. But for development purposes, you can use a shared file system or a local file system. And it also supports emerging support for Azure and Google Cloud. Okay, so in terms of understanding, does this system, I mean, it sounds great on paper, but does it actually deliver when we use it for large-scale computes? Something on the order of PCAWG or maybe even larger. And that's exactly what the group did. So about 3 months ago, we did a 20,000 RNA-seq sample recompute on Amazon EC2. And this pipeline was very straightforward. It's just using Callisto, Star, and RSIM in parallel steps here. And the data set that we were using is the TCGA data set along with GTEC, Target, and PNOC. And so I think this slide really, in a nutshell, underscores the scalability and robustness and why Toil is the step forward for us beyond what we did in PCAWG. So on the graph on the left-hand side, what you're looking at is that cyan color. That is just running the RNA-seq workflow as we did with PCAWG, single VM, running a pipeline for a particular sample. The dark blue line is showing the cost of running in Toil mode, where there's an auto-scaling cluster behind the scenes. Nodes are scaling up, nodes are scaling down. And what we're finding is we're going from about $8 a sample down to about $3.85 or so per sample. All right, so that's a huge drop by just taking advantage of the ebb and flow of computational needs for a cluster at a particular moment in time. Another step down is actually then using spot instances and taking advantage of that robustness to failure. If spot instances go away, they can be re-provisioned as the price comes back down. And with that, we're able to drop the pipeline that originally ran for about $8 a sample down to $1.30. So really just doing some very simple things, not rewriting the pipeline, not fundamentally changing the infrastructure that we're using, we're able to really drop the price down. On the right-hand side, what I'm showing is the cores used over time. And the two red circles indicate where the spot marketplace spiked, and we lost all of our worker nodes. So what's happening here is the system is recovering from that. It's launching worker nodes again, and it's picking up where it left off, ultimately to hit about 32,000 cores managed by Toil. What I think is really cool is in PCAWG, it took us over a year to do the analysis. In this effort, it was about 4 days to do the 20,000 because we could really spike our usage in Amazon and really take advantage of the scalability of infrastructure like S3. All right, so I gave you an idea of what PCAWG showed the community. I think it showed a lot of cancer researchers that we could use the power of commercial clouds for large-scale scientific work. I think that was a fundamental thing that came out of the PCAWG project. Our current work on Redwood, Dockstore, and Toil really formalized the lessons we've learned, and we've tried to translate what we've learned from PCAWG into things that other people can use, that the community can pick up and use right now. Our future work is to extend that even farther by focusing on how can we establish standards for Redwood, Dockstore, and Toil so other groups, other efforts can implement similar systems but provide common APIs. So I can use Toil and I can use other systems in a common way. So that's our current focus for our future work here. And this is all really being focused on by the GA4GH. So this is the Global Alliance for Genomics and Health. This is where our API standards effort is taking place. So I'm the co-leader of the Containers and Workflows task team. And our priority there is to establish APIs that abstract and make generic the facilities of Dockstore and Toil and these other pieces of infrastructure. So the first one we've worked on is the tool registry API. And this is just a very generic way of describing this Docker image is available. Here's its descriptor. Here's its source in Quay.io. This API, we hope that multiple tools like Dockstore will support it. And that way we can essentially have a federated network of GA4GH tool registry supporting systems so we can share tools and workflows in a standardized way. So Dockstore currently supports it, and we're working with BioShadok and others for those repositories of Docker-based tools to essentially expose their tools to the world using this API. So another API that we're working on through the GA4GH is the workflow and task execution APIs. And these APIs are essentially the same API under the hood, but what they're allowing us to do is to take a WDL or CWL workflow or a Docker-based tool and actually make a request to a system that supports the API to then run that tool with the parameters we've provided. As a result, we get back a status URL and then can retrieve file outputs, locations, as well as standard error, standard out, the status, and that sort of thing. And so this is an area of active development right now to establish this API standard. We're hoping to have the first version of this API schema released in early 2017. As far as the tool registry API, that's already released as a 1.0 schema release as of last month. So where are we headed? I would love to have had this infrastructure, these standards, Dockstore, Toil, Redwood. I would love to have had those 2 years ago when we worked on PCAWG and we really scaled out our infrastructure and built out so many different tools. In the future, though, I think establishing standards on top of things like Dockstore, Toil, the storage systems, establishing GA4GH standards on top of these systems, I think is really important because it'll enable future systems, future large-scale distributed cancer analysis projects like ICGC Med to access a variety of tool registry, workflow registry sites like Dockstore or Bioshadok. It would allow us to schedule out workflows in a standardized way to, say, Toil or Consonance, FireCloud, or Seven Bridges. And those individual steps can then run in a variety of cloud environments using the task execution API, driven by things such as cost and also location of data. So we're hoping that the GA4GH standards enable this sort of view of the world going forward in the future. And we're working closely with a lot of these organizations. This is not necessarily a commitment from these organizations, but more of a vision of how this might work in the future. So with that, I just want to thank a ton of people with the GA4GH, in particular Lincoln, Josh, Gaddy, Peter, and Jan for leading the technical working group of PCAWG, Vincent for the storage infrastructure, Dennis for Dockstore, Kyle for the task API, Peter for the workflow API, Jeff for the co-leader of the GA4GH task team, and Hannes and Frank and the Toil team for Toil infrastructure. I do want to mention that these projects are all open source. We welcome your contributions. We welcome your use of these projects. We want people to use these. We want people to benefit from the infrastructure that we've built for PCAWG and infrastructure inspired by what we did in PCAWG. So for more information about Redwood, check out the ICGC DCC Get repository. Dockstore is in production right now. You can use it, find all of our documentation on dockstore.org. And Toil is something that you can spin up today, write workflows locally on your laptop, and transition those to AWS, following the guides on our Read the Docs. At this point, I'm going to hand it over to Angel Pizarro to get the perspective from AWS.
Years of work. Really fantastic results. I love seeing the progression of good projects like that develop into something that the whole community can use. So kudos to all of the international scientists doing that work. My name is Angel Pizarro. I work with the research and technical computing team focusing on genomics and life sciences here at AWS. Work with folks across the globe on these types of problems. And given the backdrop that you just saw, I want to give why AWS cares about these problems, what our perspective is, and sort of directions of why we do what we do, right? So enabling science. I think nothing is more important to AWS than changing the world, and science is one of those areas that we want to change for the better. And for the previous talk, I hope you could recognize some of these features that deploying to AWS enabled. Specifically, the scaling of compute resources only when they were needed to when they're actually needed, drastically reducing the time to a result, because in science, really any single result is not as important as what comes after it, and what you do with those results and how you integrate those results with other data. Of course, cost was greatly reduced, and I cannot emphasize enough what a game changer is to be able to securely share data in place for some of these large cohorts. The download and compute model worked up to a certain point in the life sciences. It doesn't work anymore when your data sets are literally petabytes of data. So being able to securely share data in place, bring the compute to the data is a huge change for science and one for the better. And also having a global community being able to access that data in the same way should not be overlooked as well. So we have an open data program, and this is how we see the world. We really think that data is critical infrastructure for a lot of different types of sciences and businesses and software development and ISVs. Again, if your project depends on a petabyte of data to actually work, then data is a critical infrastructure piece of what you're trying to do. For instance, the EXACT database proved, they had a very interesting paper about being able to look at cancer data and only sequence the tumor as opposed to a tumor and normal tissue so that you could see the differences. With this large backing database from a population of people, you can actually reconstruct what would be different if you had sequenced the normal tissue. And that matters because you've essentially cut the cost of sequencing a cancer genome in half. And that's really, really going to matter as we start getting genome sequencing integrated within the clinic. So again, we believe open data is critical infrastructure. And to date, the public datasets program, I'll use the mouse, is really here, where we're creating a place to put data at rest and making it available in place. The work that Brian just showed is in this area where you have data catalogs, you have basic APIs to access that data, and the tools and visualizations to make that available. We have a little bit of complex APIs currently under development for working with the data, and so on and so forth. So where are we with bioinformatics? Of course, we have data generation taken care of pretty well. We can produce a hell of a lot of data with the current instrumentation that's in place, whether that's Celexa-based sequencing or the newer nanopore sequencers that are coming out. And then, as we showed with the ICGC dataset and the PCAWG dataset, you can make that those types of data available at rest. We have groups like the Global Alliance really making basic APIs that enable folks to talk in the same language and access the data in standard ways. And then you have data visualizations like CBio Portal and data distribution frameworks like the national repositories like NIH and the EBI over in Europe. Visualization, we do have some pretty good tools. You know, the genome browser from UCSC is one of the granddaddy tools. It's been out for 12 years, if not more. And it's still highly developed, highly used by the community. The rest of this, though, is really work that's under development. Predictive modeling, we're not there yet for genomics and life sciences, I'll be frank. Complex APIs, there's a few of them, but there's not really a good sense of what is capable and doable. Consumer applications, you're starting to get different applications of genomics in the consumer space, but how useful that is still an open question. Data-driven journals, like can we create public policy based off of genomic data that's coming in? For instance, if you're doing ubiquitous sequencing of sewers across a city, can you spot food deserts by sequencing the poo? Maybe. I think that would be pretty cool. Free idea. So, you know, Amazon S3, I'll refer to the previous talk, LFS304, about creating data lake architectures for science space. But we see S3 as a critical component because you can start storing not only genomic data, but data that's coming out of your electronic medical record, data that's coming out of your family history, from our smart connected devices, from more regulated workloads. Can you do passive monitoring of people, integrate that all within a common data platform, and then start bringing in tools from either machine learning or classical statistics to develop new models of ongoing care and providing care. And obviously, visualization is a good place. If you did miss the last session, 303, Life Science 303. It's actually going to be repeated tomorrow at 3:30. So look for it in your schedule. As I mentioned, the public datasets program does have the Cancer Genome Atlas and the ICGC cancer datasets. Those are two separate petabyte scale datasets. We're working with the National Cancer Institute to make more data available. We also have some reference public data. So if you're an algorithm developer and you don't, TCGA and ICGC are both controlled access, right? They are derived from humans. They are consented for research, but obviously we don't want folks to do bad things with that data. So there's a minimal blocker to make sure that you're not a nefarious person. But we do have reference data sets that are out there in the public domain, including the 1,000 Genomes project, which has actually 2,500 genomes, so it's kind of a misnomer. A genome in a bottle, as well as a human microbiome project, and also getting into the crop sciences as part of the public datasets program. We also have earth sciences and other data types for internet and social sciences, like tax forms. So if that's your thing. But that's the picture with data. That's the picture with open data as a platform. We saw that providing that data on scalable systems like AWS does lead to different architectures than your traditional HPC cluster that has a shared file system, a scheduler, a fixed size resource that you have to contend with other users. We saw over the years refinement of the computational model to discrete applications and workflows that distribute processes on a per job basis. So what else is out there? What is the future for bioinformatics and life sciences? And I'd like to discuss a little bit today about serverless science. Again, I'll refer to a different session earlier today that goes into much more depth about using Lambda for control systems. That's Life Sciences 301. You can catch it on YouTube. But today we're going to talk about a few other ones. So this is a little bit slightly out of context. I don't have enough time to really delve into the details of AWS Lambda, which is our serverless event-driven compute service. But the gist is you write a piece of functionality, a very small piece of functionality, and don't have to manage the servers to run that piece of functionality. It's a microservice. And AWS itself will handle the continuous scaling of that resource for you and bill you at sub-second metering. The key scenarios for AWS Lambda are data processing, where you've got stateless processing of discrete data as it's coming in. You have back-end development systems, so being able to execute server-side back-end processes across the platform. And you also have control systems that react to data as the system changes, right? So what does that look like for bioinformatics? We have this on GitHub, our AWS Labs site. We do have a CloudFormation template and a walkthrough that shows you how to do image thumbnail processing based off of data that goes into S3. So we have CloudFormation templates that you send in an image. That image will trigger a SNS event, and we have Lambda functions listening for that SNS event that will create a thumbnail and put that thumbnail back in S3. And I said, well, that's actually pretty close to how genomics processes work with Oxford Nano, right? Oxford Nano being a small thumb drive sequencer. You get data as it's coming off of the instrument in real time, and you have to call the DNA bases that you saw from event data, so electrical signals. There's an open source caller out of Jared Simpson's lab called NanoCall. So I took the CloudFormation script, I forked a repository. The hardest thing about this was actually statically compiling NanoCall so I could wrap it up in a zip and send it off to Lambda. But it actually works, and it took a few minutes of work. And now you have a resilient, scalable system for dealing with real-time genomic data. Again, if you have an air filter that's looking for specific pathogens that might be brought into an environment, you actually have a pretty simple, scalable system to address those needs. Another case study, so we're looking at backend data processing. Station X's gene pool platform. This is a case study that we recently released a few months ago. I believe that they're at the Converge Health booth, so if you want to talk to Station X, please feel to reach out to them. But they essentially created they wrapped a R function and statically compiled R within Lambda. So this is a rather large program. But they were able to fit that within a Lambda function and use their control plane, which is PHI compliant, segment out the data that's not PHI, send it in through to Lambda, and then get the results back. And now they don't have to run a fleet of dedicated servers, which is a much larger cost and much less performant than having it scale on an as-needed basis. So this is a pretty interesting one. You can read up more about it. But it shows you hybrid architectures of still having EC2 environment as your main data platform with a serverless environment that is handling data at scale. So pretty interesting. And if you want to go full control system, all Lambda group, Ciro out of Australia, just released a complete serverless data processing for CRISPR-Cas9 searches, right? And what's great about this is A, there is literally no servers in this. It's completely a serverless environment. And they did some really interesting things because they're doing whole genome scans. And obviously, Lambda is a resource-constrained algorithm. So you have to segment the search database into lots of different Lambda functions based off of the parameters that you get in. So the way they did it is they took a candidate CRISPR site, they did an immediate search for the template scan and gave it a score, and then returned that result to the user. And in the back end, they said, okay, I am going to create a Lambda function to create SNS topics to do the full genome scan so we get more resilient information on that CRISPR search that would give you off-target effects or other likely sites that might be better, right? And then all of those results go back into DynamoDB, and we have a polling process within this static S3 site that will update your result as these functions finish. So very, very cool architecture. I think we're going to see a lot of different architectures using Lambda within bioinformatics, and pretty cool, innovative work. So that's all I have for you today. I want to thank Brian. I want to thank the entire Research International community for making that resource available. If you guys have any questions for us, if you want to dive deeper into any of these subjects, please save your questions. We'll be in the back of the room. And have a good reInvent. Thank you.